Good afternoon, everyone. Thank you for joining this session. I also want to thank the organizers for inviting me to present on this interesting educational topic. So from Molly's talk, we have already learned that one can extract lots of meaningful information from various physiological measures. However, in certain applications, these physio signals may also confound our interpretations of neuronal activity measured by fMRI. So the focus of this talk is to discuss some potential approaches to mitigate various noise incurred by physio processes in order to improve the neuronal specificity of fMRI. As we already know, fMRI directly tracks neural activity through the cascading hemodynamic changes. And we learned from the previous talk that accompany these neurogenic responses triggered by external or internal stimuli, there are also active autonomic processes going on in the body. And changes in respiration, heart rate, or blood pressure may also incur blood oxygenation level changes at a range of temporal scales. And vessel and body movements coupled to these processes may also incur magnetic field changes that modulate the signal intensities or directly disturb the dynamics of functional measures. And additionally, these autonomic processes may also interact with neuronal state changes. So in order to infer truthful neuronal information from fMRI measures, we have to address all these physiological noise. So for my talk, I will first introduce how we can retrospectively correct for these physio noise at different temporal scales, and to separate my mission from the talk by Cesar, I will focus on the denoising approaches based on external physiological recordings. And I will then highlight some additional considerations on the acquisition side to mitigate noise. And finally, I will highlight some challenges of denoising when these autonomic signals track neuronal state changes, and also highlight some potential avenues to correct for physio noise in these situations. Okay, so I first will introduce some methods to mitigate noise time locked to cardiac and respiratory cycles. So if you inspect the raw fMRI images over time, you can already notice some pseudo-periodical changes of the signal intensity surrounding those big muscles, for instance, highlighted by the red arrows here. And these changes are more obvious if you focus on those ventricles or the sagittal sinus areas. So what are the causes for these types of quasi-periodical motion or intensity changes in fMRI signals? On one hand, this cardiovascular pulsatility will cause motion that result in dynamic partial volume effects in fMRI signals. And on the other hand, these physio processes will also cause periodical variations in blood oxygenation level that can translate to fMRI signal intensity changes. So for signal variations incurred by these two mechanisms are typically localized to big vessels. For instance, here plots the spatial distribution estimated by the Human Connectome project data. As you can see, those big vessel area has the most influence from these cyclic physio factors. And in particular, for instance, for some regions like insula or brainstem, like they are in the vicinity of those big vessels, so the contamination is kind of more severe, and we should be quite careful about it. And this may also arise from head motion coupled to respiration, and the pattern is more complicated depending on the specific types of motion the subject have. And another cause for this fast physio change is a field perturbation caused by chest movements coupled to respiration. For instance, in this nice example, you can clearly see that the face image of the neck and brain kind of co-vary with the magnitudes of the respiratory traces. So for this type of noise, it has a very clear spatial signature that its influence will decay as the distance from the chest increases. For instance, in this example, if you plot the phase shifts as a function of the brain area, you can see the brainstem area has the most influence. Okay, so after learning the different causes for these periodical physio noise and how can we mitigate them from fMRI measures? So as both respiration and cardiac signals reside at frequencies that are much higher than neurogenic fMRI responses. So if we're able to sample faster enough, we can directly notch filter in this influence. But due to the presence of those higher harmonics, especially also for the cardiac traces, you have to sample really fast to account for these changes. So in typical cases, these higher frequency fluctuations can alias into lower frequency bands and overlapping with the spectra of those interesting neuronal fluctuations. So in this case, we will be able to infer or estimate these noisy physio waveforms from external physiological recordings. Okay, respiratory is probably the most widely used modeling approach to account for cardiac and respiratory noise in fMRI time course. So this approach models projected physio noise according to the specific imaging phase within each cardiac and respiratory cycle. So for instance, we use a cardiac cycle as an example. We have those pulse measures, and so you detect those cardiac peaks, and here's your imaging volume. So for each volume, you infer the associated phase based on the timing within each cardiac cycle. And after obtaining these phases, you can expand it into higher order series and to model these cardiac signals. And respiration phase can be inferred in similar manner, but there is a little bit more complicated process in accounting for the gating in the signal magnitudes at the beginning. So I wouldn't go into that details. And for the selection of order of the Fourier series, most studies will select an order higher than 2. For instance, as shown in the group level characterization here, there is still a considerable variance explained by the second order series. And sometimes we may also consider adding an interaction term to account for potential coupling between respiration and cardiac processes through mechanisms such as respiratory sinus arrhythmia. This is typically realized by adding a multiplicative term of the respiratory series and the cardiac series. And here it shows an example where the interaction term accounts for additional variance in the velocity of brainstem. So how can we determine whether we need to have these additional terms for denoising purposes? It's hard to say. You probably can only see the significance of the covariate after including them in. But sometimes if you sample fast enough, you can clearly see those signal peaks different from the fundamental harmonics of cardiac and respiratory traces. That might indicate there is interaction there. Okay, so not just doing the correction in the image domain, it's also possible to perform correction in the raw case-based data before reconstruction that called retrocacal. But retrocacal is not very common nowadays due to various reasons. On one end, when you perform correction in the case space, so it will be dominated by those lower frequency structure patterns, so that resulting in the image that the higher frequency details are not denoisable. And also because nowadays we're using multiple coil acquisitions, if you perform correction in the case space, it will incur very high computational loads. However, there has been some arguments stating for those long TR volumetric acquisition, where the inherent assumption of simultaneous acquisition and image of rhetorical is violated. Rhetorical could be something to consider. Okay, so after modeling this physio noise, the way to project ultimate data is just using linear regression. So this is very standard. But I want to highlight a potential caveat here when we're having these respiratory artifacts in motion traces. Sometimes you can directly see the coupling between certain motion direction with this respiratory waveform. They may not arise from respiration coupled head movements. Instead, they may be caused by those field perturbations induced by chest expansion related to respiration. And an easy way to identify is that this change will be most prominent in the phase encoding direction. And because these traces doesn't represent true motion in the head. So if you don't address them using these measured motion traces to denoise the data, the effects may not be effective. So there has been some work out recommending addressing these physical artifacts in the motion traces before denoising. And actually one simple way to do this is just to include the respiratory traces together with motion in the denoising process. Okay, so next we will focus on those physiological noise occurring at much slower scales. So this will be kind of include those very slow variations in the end tidal CO2 level or respiration volume, because they both relate to because CO2 is a potential vasodilator, it will modulate the blood flow, and also blood pressure or variability in heart rate, and kind of modulate the cardiac outflow as well. And then because they will cause signal changes from similar mechanisms as the neurogenic responses, basically blood oxygenation level dependent mechanisms. So it's very hard to tease them apart from those neuronal fluctuations of interest. So if you look at their spectrum, unlike the noise we introduced in the first section, they really overlap with this spontaneous brain activity. And if you look at the spatial patterns, they also confound the gray matter voxels. So for instance, this is a seminal study where they correlate the respiration volume per unit time, kind of seeing the blue curve here, the envelope of these respiratory traces, and it correlate to global fMRI signals. And you can see it surprisingly explains the majority of the variance in many gray matter areas. And here is another example where they correlate the heart rate variability to region-wise fMRI time course at different temporal delays. And you can also see prominent correlations between these autonomic process and fMRI signals. So how can we correct for this physio noise? And this is typically done by model these responses by convolving these physio measures with a canonical physio response function. And here are some examples of common functions established by earlier studies. For instance, including the respiration volume per unit time or variation in the respiratory magnitudes over time, or heart rate variability, or end tidal CO2 level, and each associated with some characterized canonical functions by earlier studies. And you can notice some similarity with the canonical hemodynamic response function linked to neural activity, but the timing has some slight changes. But for some newer physio processes, such as the amplitude of pulse waves or the blood pressure, this transfer function or physio responses remain to be established. And here just shows one example, kind of showing the effects of removing these slower scale physio changes, because we're roughly kind of coherent across the brain. So removing it will kind of being able to resolve some anti-correlations in the brain. For instance, here it shows the functional connectivity with respect to a seed in the posterior cingulate cortex. As you can see, after correcting for these slow variations in physio noise, you're going to see clear anti-correlation patterns, also diminished spurious positive correlations. And also because different physio sources reflect distinct aspects of the autonomic regulation, while they are to some extent coupled, they do account for complementary information in the fMRI dynamics. And the fractional contributions vary depending on the specific scanning experience of each subject. For instance, as shown by the example here, like there are different composition of these variants from different subjects. So that means if you have access to different physio sources, you should consider include all of them in the noise model. Okay, while these physio dynamics are roughly coherent across the brain, they do demonstrate some local variations. This is the earlier study by Rasmus Bern. They already showed in the fMRI dynamics coupled to respiratory volume per unit time, there is a temporal delay across the brain. And this is not hard to, or it's not quite surprising because we know that our brain has a very heterogeneous vascular tree across the brain. So when systemic physiology kind of filters through this complicated vascular tree, and you likely will observe regional differences in the both responses. So in an earlier study, we used a human connectome project data to characterize the regional variability of these responses. So in this example, here shows the global fMRI dynamics following an increase in respiratory variation. And here shows the global fMRI changes following a decrease in heart rate. And you can both see that they're quite variable across the brain. And not just regional variability of these responses, these physio changes are also variable across subjects. So here shows an example of characterized cardiac response and respiratory response functions from two different subjects, and you can see they're not quite consistent. And if you plot the variance explained by the group level physio response and individual physio response, you can see using the individual response as a regressor can significantly increase the variance explained by these physio processes. And also the response may vary according to the TR you use. So actually several studies have actually cautioned that these physio response function may differ between different TRs. So there are many explanations for this. One possibility is that there might be slightly different contrast mechanisms associated with different TR. For instance, when you shorten TR, you reduce the signal to noise ratio and also have increased inflow effects to the collected images. And another possibility relates to the sampling frequency or sampling variability. So for instance, if you have longer TR, you sample the data at longer intervals and you come out with a kind of delayed response. But when you have a shorter TR, you actually sample the traces at faster scales. And to achieve the same measurements, they were going to have a shorter TRs here. So how can we account for this spatial, individual, or TR-dependent variability of these physio responses? There are many ways to do this. For instance, some studies will first do a cross-correlation to determine the relative lag between physio traces and voxel-wise fMRI responses, and then use the optimal delay to account for variance in the data. And you can also consider adding a temporal or dispersive derivatives just to account for this regional variability or individual variability of these physio changes. And you can even push it further using more flexible basis functions, even the finite impulse responses to account for physio changes if you have lots of data sets that degree of freedom is not a primary concern for your data. Okay, so what I've mentioned earlier, how we can retrospective correcting for this noise using external physio recordings. I just want to highlight in one particular aspects of data acquisition that can help us mitigate the physio noise. So for commercial acquisition, we're typically using very large voxel sizes, but actually going to smaller voxel sizes may be helpful too. For instance, for big voxel, you always have contributions from those big PO vessels, more vulnerable to this physio noise. But by using smaller voxels, we can sample data from the parenchymal regions in order to mitigate the contamination from physio sources. And similarly, for those slower scale changes, there have been many studies showing that by pushing the data to the white noise dominated regime just through this, reducing the voxel size, we can mitigate influence from physio changes. And additionally, there are also reasons like these physio changes are still in general more coherent compared to potentially neuronal fluctuations. So then if you have a big voxels and there will be white noise cancellation or even some local neuronal activity cancellation, but the resulting magnitudes in big voxels are much smaller compared to the aggregated response of these physio changes. So here is just an example where we simulate the effects by smoothing the data. And then you can see that as you have more intensive smoothing of the data, mimicking those larger voxel cases, you're going to get a much higher fractional variance of physio noise explained by external recordings. Okay, so finally, I will just highlight some particular challenges when these autonomic signals track neuronal state changes, and then discuss some possible ways to consider addressing this noise when this situation occurs. So here are some scenarios when systemic physiology and neuronal states may co-vary, making it very challenging to account for physio noise in fMRI data. So for instance, there may be tight coupling between fMRI time course with these traces when there is a task coupled breathing patterns. For instance, here in this decision task, you can see that the respiration volume per unit time has a tight alignment with the task paradigm. So as a result, if we plot the task activation, you can see many brain regions are activated. And these effects don't seem to be specific to a particular task. And in a recent study where the authors kind of correlate the respiration volume per unit time with the task model response across a range of cognitive tasks in the human connectome project. And it seems like in most paradigms, you can see a coupling between these physio and task responses, suggesting this concern kind of may be more common than what we have expected. And sometimes the task is also designed to heavily engage autonomic nervous system. For instance, here is an example that studies the brain activation when Tai Chi masters concentrate on their Qi. And in this case, there is a tight coupling between breathing and the task. So most of the neural activation are gone if you perform the physio noise correction. So we should be very careful with physio noise correction in all these kind of studies, including pain or meditation. And also, brain arousal variations may also be accompanied by changes in the autonomic states. For instance, as shown in this study, the coupling between different physio processes and fMRI signals seems to be stronger in low vigilance states than high vigilance determined by EEG signatures. And here is another example where the subject falls asleep. And you can see that in stage N2 sleep, every time there is a K-complex event, it's accompanied by a dip in the magnitudes of the pulse oximetry. And both these neuronal measures and physio measures are coupled to the global fMRI signal change. And I really don't want to make the issue sound too complicated, but in any situation, these physio processes are still neuronal in origin. So some brain regions are always actively involved in this autonomic processing. And changes in autonomic processes may also reversely modulate respiration. For instance, here is a study that shows respiration can modulate the amplitudes or phases of different brain rhythms. So what can we do when autonomic states co-vary with neuronal states, and what are the potential avenues for us to disentangle or identify the dominant neural or physiological sources? And animal studies or multimodal imaging are always reasonable directions to go if we have access to more direct measures of neural activity through optical or electrophysiological measures. It's always a good idea to help us disentangle these physio and local neuronal effects. But here, I want to highlight some potential thoughts on how to tackle these questions in human fMRI. These are just preliminary ideas. So first, probably information from the deep brain structures may provide some insights. For instance, if you contrast the fMRI spatial patterns of arousal effects and regional respiratory variation, while the spatial maps are very similar in the cortical region as shown here, the behaviors of thalamus are quite different. It seems to be more engaged in the arousal states. So the idea is that it's very natural for us to expect quite distinct activity patterns in subcortex and brainstem associated with neuronal state changes or autonomic states. And this is still an under-investigated domain, but has lots of potential. And second, cortical layer analysis enabled by recent advances in high spatial resolution fMRI may also shed some new insights into this puzzle. So it has been established that for neurogenic responses, this neural activity occurs in the parenchymal regions and then propagates downstream to the PL surface, while for changes in fMRI dynamics incurred by systemic physiology, the temporal order is reversed. So it's starting from PL vessels and propagating to those smaller vessels here. So resolving these timing order differences in future studies may also share some insights into these differences. And not just seeking for additional information from the space domain, timing in the associated fMRI responses may also be informative. So you might have already noticed that these physio responses and neurogenic hemodynamic response function have very distinct timing. So for local neurogenic responses, it may take 4 to 6 seconds for the signal to peak, while for these physiodynamics, it will take much longer lag. So this recent study basically used this distinction to infer the dominant drive of fMRI signal changes in stage N2 sleep and the observation of a longer delay relative to slower wave activity, such as a larger contribution from physiological sources in this state. Okay, finally, we also provide a small exercise for you to gain some hands-on experience with denoising fMRI data using model-based approach. You can download the toy dataset from the shared Dropbox link at the bottom. And in this example, we will assess the influence of physio denoising on positive and negative correlations with the seed in the posterior cingulate cortex, basically to reproduce the findings by previous studies. So after loading the four-dimensional fMRI data in seed masks, you will do a simple pre-processing to remove the low-frequency scanner drifts, and you can plot the correlation maps centered at the seed without any denoising. So here you just include different regressors associated with motion and physiological noise modeled at both faster and slower scales, and you're going to assess their influence on the derived functional connectivity patterns. And if you run through the scripts, you will get the following connectivity results. As you regress out more of those physio noise, you are supposed to get more cleaner representations of brain connectivity. For instance, those negative correlations become more stronger compared to the raw data. And also you see those kind of posterior parts of this positive correlation disappeared after denoising. So hope this will give you an intuitive demonstration that physio artifacts can significantly alter the functional connectivity measures, and it's very important to account for these changes when deriving truthful measures of brain neuron activity. And here are some very nice review articles on this topic if you want to learn more about mitigating physio noise in fMRI time course. Okay, so now you may wonder, after knowing all this approach, which software or toolbox you can use to implement these model-based denoising approaches? And what if these external recordings are missing or have very poor quality? And can we mitigate noise from the inherent information from the data itself? And finally, while here we view these physiodynamics as noise that may confound interpretations of neurogenic responses, we can also leverage the rich information extracted from the physio processes to infer cardiovascular health in both task and resting state sessions. So in the following talks, my fellow speakers will touch upon these different aspects. Okay, thanks for your attention.